Welcome back to topic 7, which is all about nucleic acids. Today we are going to dive into DNA replication and really sort of just talk about the enzymes you need to know how how DNA replicates, and how we can sequence DNA strands. So let's talk about the basics before we dive into anything else. The picture in the top right corner should bring back these hidden memories, hopefully, of the cell cycle, which is how cells replicate. The G1, and G2 phase are all happening during interphase, and DNA replication in particular happens during the S phase of interphase, right? The S stands for synthesis, so that's when replication happens. So this produces sister chromatids, which will then be separated in mitosis, and then when the cell divides, each daughter cell gets a full set of DNA, and they are able to do this because you have enough of the sister chromatids that are produced during synthesis to be able to divide evenly into all of the daughter cells. Now, when we first started thinking about how DNA replicates, started thinking about the cell cycle, started thinking about how DNA is able to be passed on from generation to generation, there were 3 possible ways that DNA was thought to replicate. There was conservative, meaning that there was the original parent, right? Original parent, and then that original parent just continuously stayed together, and then you would get a new version of DNA, right? The DNA that's replicated, and then that parent would replicate again, but it would always stay together. So this red original strands of DNA would always be together, whereas the new blue strands of DNA would continuously be replicated. You'll notice that it says 15N and 14N DNA. We will talk about that in a slide or two, but this is how they were able to do the tests. New scientist alert, you're going to have to know a new scientist soon. They're semi-conservative, which if you know anything about DNA, you know that this is actually how DNA replicates, where the new strands divide and then you build the parent strands divide and you build your new DNA off of the parent strand template, and then they continue to divide, and the parent strands are used as a template in the new generations. And then there was also the idea of dispersive, where some of the original parent strand was used in different areas, and you can kind of see that it's just like all a mishmash throughout. Of course, this is an idea. Looking back now, you're probably thinking, wow, that's silly. Like, why would that seem so complicated? And yes, of course it's complicated, but we didn't know any better. As scientists, we had no idea. All of this information came out in the last 50 years. We just sort of found out about this in the span of how time works. This is all relatively recent. So again, replication basics. We know that DNA replication is semi-conservative. So when you replicate DNA, you have a parent strand that splits open, and then you create a complementary strand, which is the new version of the strand that is produced. And so your end result is a DNA strand that is half old, half parental, and half new, half copied. And the reason we know this is, your scientist, Messelson-Stahl. So, scientists. So this was an experiment that was done. They grew E. coli to have a heavier version of nitrogen, right, N15. And that's what you saw in that picture 2 slides ago, where you had the red and the blue. You were able to see that some were N15, some were N14. So they specifically made E. coli with a heavier version of nitrogen in the DNA, and then they removed that DNA and let it replicate with the normally found version of nitrogen, which is N14 in DNA. So they had their original parental strand would be heavier, and then they let it replicate with the normal version of DNA that was found in the E. coli cells, and then they would periodically measure the density to determine how DNA was actually replicating, and then you could see if it was conservative, semi-conservative, or dispersive. And of course we know that it is in fact semi-conservative, so this is again just an example of their experiment. And so they had all of the original parental DNA as this heavier density, and so it started there, and then during their first replication, you can see that it's starting to split into half old and half new. And then as it continues to replicate, you can see that the lighter version of DNA becomes much more frequent, and that is because there's only one original parental strand of DNA. And then as they continuously replicate, the new version, like say you have parentals and then you have like the F1 generation, that F1 generation replicates again, but the half of the F1 generation is with the N14 density DNA. So when that replicates, it's just another N14 DNA. And so you have, so you have that lighter DNA throughout at a much more frequent amount. So DNA replication, we've talked about this, but DNA replication moves in a 5' to 3' direction, and we've talked about what 5' and 3' means, right? 5' is the carbon that's closest to the 5' location in the sugar, and then 3' is the third carbon in this sugar ring. So that means that the 5' end of the new strand binds to the 3' carbon of the last nucleotide to join the new strand. So it just means that it's going to, like this 5' is going to join this 3', and that's how it's going to continuously replicate. So the 5' joins the 3'. What this unfortunately does is create two types of strands, leading and lagging strands. So because replication can only occur in a 5' to 3', when you unwrap the DNA, right, you untwist the DNA, you have anti-parallel sides. One is going to go in that 5' to 3' direction, but one is going to go the opposite way. So in the leading strand, and you can see that here, it's going to follow and it's just going to continuously be able to replicate as it goes through. It doesn't need to stop because it will always have this new area to replicate on, because as the DNA untwists, the new nucleotides can just join right on. But the lagging strand cannot do that. So what happens is you start with your RNA primer, and it will start in a 5' to 3' direction, but eventually it's going to hit the end of the DNA that you're replicating. And so it'll stop, and then it'll go back because the DNA has untwisted more, so now there's a new area that they can replicate, and it'll put a new RNA primer, and it'll create another little tiny fragment until it hits that piece that you've already replicated, and then it'll let go, and then it'll make a new area to join on, and then it'll make another little tiny fragment, and it'll keep going and going, and that's called the lagging strand because it's all just disjointed little tiny fragments, and those fragments are called Okazaki fragments, again named after the scientist that discovered them. So more scientists for you to know. But the big thing is leading is continuous and lagging is disjointed. So now let's talk about all those important enzymes that you guys are going to need to know. And yes, you will need to know how DNA replication happens, with specifically referencing all of these enzymes. So you've got DNA helicase, DNA gyrase, and single-stranded binding, or SSB proteins. So helicase unzips the DNA, right? So DNA is typically twisted in a double helix, and it rips it apart, basically. Gyrase reduces that strain when you're unwinding the DNA, because remember that DNA is supercoiled. So if you just untwist it, if you've ever seen those like wicked old like rotary phones that have the cord and they get all twisted up, and when you like pull it open, it wants to twist the other way, that's basically what gyrase prevents it from happening. It prevents the over twisting and reduces the strain so that you can kind of have a nice and smooth DNA replication process. And then SSB proteins bind to the DNA strands after they've been separated so that they don't reconnect, keeping it open to be able to go through the entire process of replication. And then the other ones you need to know are RNA primase, DNA polymerase III, DNA polymerase I, and DNA ligase. So RNA primase is where you have a short RNA primer, and that's the initiation point for DNA polymerase III. So RNA primase is like your initial touch point on the DNA that's being replicated, and once the RNA primase goes down, then DNA polymerase 3 comes in and can join free nucleotides together in that 5' to 3' direction. Once DNA polymerase 3 comes in and puts all those nucleotides together, DNA polymerase 1 comes in and removes those RNA primers from the lagging strand and replaces them with DNA nucleotides. Yeah, I know. This is like, it's complicated. Of course it is. Why wouldn't it be? And then DNA ligase is like your final step on that lagging strand, and it comes together and it joins all those Okazaki fragments together by rejoining those backbones together to make it a continuous strand. This is a lovely photo that just sort of helps explain exactly what's happening. So again, you have your helicase, which is unwinding it, your gyrase, which is lessening the tension. You have your SSB proteins keeping it pulled apart. And then you have your, where is it? I guess this is your RNA primase. It makes your primer, and then polymerase 3 extends the chain, polymerase 1 removes the primer, DNA ligase finishes off that bond. Now let's talk about how this actually starts. So DNA replication in eukaryotes, which is what you are, right? You're a eukaryote, you have a nucleus, is initiated at many, many points in eukaryotes. So it just makes it faster and it makes it more efficient. These are called origins of initiation. So that makes sense, right? Because an origin is the beginning, right? So the beginning of the initiation of DNA replication. So there are proteins that will bind at these origins of initiation, and then DNA helicase will attach, and that's how you're able to actually start the whole process. In prokaryotes, though, replication is bidirectional. That's a little different than in eukaryotes, right? We go in one direction, right? We've been talking about this the whole time. And this is because prokaryotic DNA is looped, right? It's a circle, where eukaryotic DNA is just long strands. So in prokaryotes, you have one initiation point, and then it just replicates in both directions from there. And then last but not least, we're going to talk about sequencing. And the big way that we've done sequencing is using the Sanger method. So using the Sanger method is a way that we have attached these fluorescent dideoxynucleotides, and they prevent elongation of a nucleotide chain. So basically they stop replication as soon as they hit where they're supposed to be, right? They're nucleotides, but they're missing a hydroxyl group. So they will connect normally in a DNA replication strand, but say they'll connect to, I don't know, they're complementary to a T, so this DDNTP-A, right, because A and T go together, will connect, and then nothing will be able to connect after it, and it will just stop replication in its tracks. And the reason that we care about this is because by putting a bunch of these DDNTPs in with the replication process, you can actually stop the replication process from happening in certain areas, and then you can sequence what that DNA that you're looking at actually is saying. So you can determine specific nucleotide positions based on where these DDNTPs ended up. And the way that you do that is you run some PCR, which is basically where you can replicate DNA a bajillion times. And I'm serious, you replicate it like many millions of times. And then these fluorescent tags, right, they're all different colors, which makes it easy to see. You will be able to determine where they stopped the replication process. Then you can separate with a gel or use a sequencing machine, and you can actually see, okay, well, this is my smallest piece of DNA, right, this T, because it moved the furthest down the gel. If it's lighter, it's smaller, and if it's lighter, it can move further. And then you'll get heavier and heavier, and you can actually see, right, this is the heaviest piece of DNA, this G that matches up with this G, which is also the heaviest piece of DNA. And you can follow it all the way through until T, which is the lightest piece of DNA, and your last piece, which is the T again. So the lightest piece of DNA. We'll practice a little bit more with Sanger sequencing. It's pretty easy once you get it, but we'll just, do a little bit of practice in class. All right, that's it for today. I will see you next time.